Hi everyone, and welcome to today's webinar entitled Plant Biology, Pitfalls and Solutions to Frequently Used Research Techniques. My name is David Lin, and I'm a technical writer / marketing specialist here at GenScript. A little bit about my research background. I did my master's degree at University of Maryland, College Park, specializing in hormone signaling in plants, specifically Arabidopsis thaliana. So let's get to it. A little bit of an overview of what's going to be covered today. First, I'm going to briefly go over some practical applications of plant science. And next, I'm going to go into the meat of the presentation, common techniques in plant research, and some of the things to consider if you're looking to go into plant research. Third, I'm going to talk about not only gene synthesis as a tool, but also specifically codon optimization and how it can help you reach your goals in plant research. And lastly, I'm going to introduce some resources that could be of value to you when undertaking this endeavor. So first, practical applications of plant science. Plant science as a whole has a lot of broad appeal these days, anywhere from helping with the investigation of alternative fuels / biofuels, improving crop yields, helping maintain ecosystem maintenance, and also several medicinal applications. So as you can see, a lot of the science done in everyday plant labs can have a wide variety and broad appeal to the general audience and society as a whole. So some common techniques used in plant biology research. One of the main ones I've seen during my time at University of Maryland is fluorescence microscopy, and furthermore also biomolecular fluorescence complementation. I've also seen a lot of co-immunoprecipitation and immunoprecipitation used, plant growth assays, and also, in my particular field, analyzing signaling pathways. So I'm going to touch on all of these bases today during the presentation and hopefully help you troubleshoot whatever issues you might be having with your own plant-related research. So with fluorescence microscopy and biomolecular fluorescence complementation, otherwise known as BIFC, there's a lot of broad applications for this technique, anywhere from identifying cells and their cellular components with a high degree of specificity, differential studies and general protein localization / co-localization, and also identification of interacting components. And this is the main purpose of our BIFC assay. So some things to consider when using fluorescence microscopy in your research workflow. One of the main issues with plant cells, and in my case I was using leaf cells in particular, was the autofluorescence seen with these cells. And so you want to make sure that your tags are compatible with the natural fluorescence seen in your host plant. Furthermore, expression levels and tag compatibility, you need to actually be able to balance your expression levels with the natural protein function so that one is not overriding the other. And I'll explain what I mean by that in the upcoming slides. And lastly, you'll want to look at protein co-localization versus interaction, and there's different techniques for each goal that you're trying to achieve. So with autofluorescence, it's defined as the natural emission of light by biological structures and organs when they have absorbed light. So some things to remember when looking at this, and what we have here on the image on the left is just autofluorescence that you can see from chloroplasts in plant cells, and with the green being the actual fluorescence of your fluorescent protein of choice. So some things to remember is compatible tags. So as you can see here, the author of this picture was able to choose a green fluorescence protein against the background of autofluorescence of red from the chloroplasts. And so this ensures that he's able to differentiate the signal from his protein over the natural signal emitted from the leaf cell itself. So what I mean by this is that the autofluorescence seen can actually overlap with your fluorescent protein spectral signal, and this could lead to some confusion as to whether or not the light emitted from your image is actually from your protein or if it's actually from your from the autofluorescence seen. And this confusion can arise from colors that are overlapping too closely on the spectral spectrum. It can also increase, your autofluorescence can also increase due to cell damage during infiltration or from extended exposure to lasers. So both of these need to be kept in mind when looking at fluorescence. So some solutions to combat autofluorescence as an issue is to, one, there's actually upgraded confocal systems that can manually subtract background autofluorescence based on the spectral profile of the autofluorescent structure that you're looking at. So in this case, it would be chloroplast. You can set the confocal system to automatically subtract that from the background, leaving only your spectral signal of choice visible. You can also consider using tags on the end of the spectrum, as mentioned before, for compatibility issues. And lastly, you can use a host system that has less native autofluorescence than your current host of choice. So in this case, my comparison is tobacco leaves, which actually has a lot of high fluorescent background, both from the technique used to infiltrate tobacco and also from the natural chloroplastine, versus onion cells, which actually, when infiltrated with their own technique, does not cause as much autofluorescence, and your fluorescence spectral signal of choice is actually more apparent. So these are all things to consider when looking, when combating autofluorescence with fluorescent microscopy. The next thing I want to talk about is expression levels of your actual fluorescent tag and subsequent tag protein. Expression of your tag proteins can sometimes be below the detection level of the associated recording camera to your microscope. And this can actually occur due to a number of reasons. And I'm not saying that this will always occur, but this can be a reason for your low signal that you might be seeing. And one of the reasons is using native promoters versus an overexpression promoter such as a 35S promoter. These 35S promoters can increase your expression in your host cell, but there are a couple things to be wary of. There could be some unintended protein function or localization due to the flooding of your system by the increased number or increased expression level of your protein. And also, due to this flooding, you can actually have a reverse effect and cause silencing of the transgene by, or the endogenous gene by native protein silencing methods. Furthermore, fusing multiple tags to your target protein can also have an effect on native protein function. So what I mean by this is if you fuse multiple GFP tags to your protein, it can make it large and unwieldy and result in the consequences listed above, such as unintended protein function and unintended localization to areas other than the natural function of that protein. So some solutions, or the solution to this, in my experience, is to plan for redundancy. So what I mean by this is just to have several lines transfected with your transgene and then analyzing these lines for the level of expression seen. And that way you can rank them based off expression and see whether or not one might be expressing too high and one might actually be expressing too low, and then you might have one that's actually just right. And you'll also want to ensure that your transgene is aptly designed to fit your research goals. So again, test, and then test again to make sure that your transgene is expressing at levels adequate for your goals. Lastly, I want to talk about what you might be looking for when you use fluorescence microscopy. So a lot of people, one of the main goals or uses for this is for protein localization, and that is to view where your protein might actually be expressed at a particular time in a leaf cell. Another use for fluorescence microscopy and is for BIFC purposes. So the difference between these two techniques is protein localization actually just mentions where your protein is at a certain amount of time, during a certain time, while BIFC can actually tell you whether or not two proteins are actually interacting at a certain time. And so the way BIFC works is you'll have your two target proteins that you're testing for interaction each fused to their own half of a fluorescence protein. And so when these proteins actually interact, it completes the full form of the fluorescence protein, and you see fluorescence seen here on the right, in the figure on the right. A couple things to note about BIFC that you'll need to consider is that the intensity of the signal will be less than 10% of what you see with intact fluorescence proteins. And so what that means is that the signal is actually going to be a little bit dimmer than expected, or actually significantly dimmer. So one way to actually increase this association is through the use of another temporary host, such as tobacco leaves or onion cells, because in this way, you're actually, based off the method of transfection, you're flooding the tobacco leaf cells or onion cells with a higher increased number or copy number of your BIFC constructs, and in that way, you actually can see an increase in fluorescence because of that. Next, I want to kind of go over immunoprecipitation and co-immunoprecipitation in the context of plant biology. So as many of you might know, immunoprecipitation refers to the process in which you can isolate a protein from a cell lysate. And so what you'll want to do is you'll start with harvesting and lysing of your cells or tissue. In this case, or in my case, my past research experience, it was with leaf tissue or whole plant tissue of Arabidopsis seedlings. And so you'll harvest and lyse these cells with any number of specific protocols, and you'll incubate your lysate with beads or magnetic or agarose, functionalized with a specific antibody for a protein of interest. This antibody will then bind the protein, and the beads are then washed to remove any non-binding proteins, and whatever protein is resulting in your mixture is what you're targeting for your protein of interest. And then you'll elute your complex or specific protein from the beads, and you'll look at it with an SDS page to see what exactly you're getting in your mixture. And so this can be used to isolate specific proteins and also protein complexes if any number of gentle cross-linking procedures are included. Some applications for this include protein isolation and purification for any number of downstream applications, such as mass spectrometry and looking at post-translational modifications. You can also use this to, once again, with co-IP, identify protein-protein interactions and also identify other members of a protein complex. Some things to consider when you use immunoprecipitation or co-IP in your research workflow for plant biology is looking at your tag protein functionality, and also look at, and this is the most important thing in my experience, is your protein isolation technique. And so what I mean by this is the manner in which you're processing your tissues. So a little bit about your tag protein functionality. The protein actually needs to be tagged to do immunoprecipitation, so that the antibody is actually able to bind. Whatever antibody is actually localized onto the beads is able to bind your protein of interest, but you still need to maintain as close to wild-type functionality as possible, so that whatever results you're getting mimics what you see naturally in the cell. So some ways to ensure that this outcome is the most favorable outcome is to use as few of tags as possible on your protein, and usually the smaller the tag, the less undesired effects you will see. And by undesired effects, I mean random localization or abnormal localization, abnormal protein function, pseudo-protein interactions, or bad misfolding of your protein of interest. It is also possible to design a construct to address a variety of experiments and questions. And through this, I mean by the use of multiple tags. And I'll explain what I mean by this in later slides. And lastly, there's also a variety of configurations possible. So don't limit yourself. And this actually goes back to my main point of advice, is to practice redundancy when you're doing your plant biology research experiments. So by redundancy, I mean, in this case, let's say you're trying to tag your CDS DNA with a his tag and a GFP tag. There are actually multiple ways to do this, and each of these configurations might lead to compatibility issues with your host cell. So if you're able to transfect all four of these into a cell of choice, you can see which one actually might result in normal wild-type function and which ones might not be. And in that case, you can isolate whichever one might be best for your research needs, versus testing each one at a time, which could waste a lot of valuable time due to your plant's long life cycle in comparison to another model organism such as E. coli or yeast. Next, I want to go over, and this is the most important thing when it comes to co-immunoprecipitation in plant cells, is your method of processing for tissue and subsequent protein isolation. So some of the keys that I found when processing plant tissue is that the integrity of your protein is key. Nothing is actually more important than maintaining your protein's either folding structure or integrity. And so you can do this by keeping conditions favorable for protein structure and conditions favorable for preventing degradation. And so some of these include a low temperature and also protease inhibitors. Usually at higher temperatures, proteins will begin to degrade at a much faster pace, and keeping your preps or your process at a lower temperature, such as in a 4-degree cold room, can actually slow down this degradation process so that you're able to isolate your protein with minimal degradation seen. Additionally, you can also include protease inhibitors to slow down this process. If you're looking at post-translational modifications downstream, for example, you'll want to consider adding phosphatase inhibitors. And so in this case, the reason I bring up phosphatase inhibitors was because in my past research, I was actually looking at phosphorylation of a specific protein. And so I would add phosphatase inhibitors just to prevent any unwanted or random removal of phosphates at key sites that I'm looking at. So that way, in my output or my results, in my case, it would have been mass spectrometry, any of the results that I would see would be some true sort of phosphorylation or versus some random removal of phosphates and subsequent possibility of missing any sites that might actually be truly phosphorylated by my kinase of interest. And lastly, you want to consider where exactly your protein is localized. So if your protein is localized in the nuclei versus the cytoplasm, this could change the method in which you're isolating your protein. So for example, if I'm looking at a transcription factor in the nucleus and I'm trying to isolate that, I might want to consider isolating my nuclei first before lysing the nuclear membrane. And the reasoning for this is actually if you were to do a total cell lysate prep with your plant cells, you might actually get some unintended consequences of the mixing of all these enzymes and proteins in places where they normally wouldn't be, and whatever protein output you get might be low in yield or might be post-translationally modified in a different manner than what's naturally seen in the wild type. So you want to consider these things when you're doing your protein preps. Next, I want to go over a quick rundown of plant growth assays and some keys to remember when doing these. So what plant growth assays refers to is growing plants in steady conditions to look at any phenotypical differences you might see between wild type or between various transgenes relative to each other. So the key to this, as you might imagine, would be to maintain level growing conditions across all of your experimental plant transgene lines. And so the way to do this would be to make sure that your transgene expression levels are level across all different transgenes and wild type levels, to keep your growth variables and conditions steady, and also to have an accurate way to quantify your output and phenotype. So a quick rundown of transgene expression levels. The main question you want to ask is what exactly are your goals that you're trying to achieve with your transgenic plants? Are you trying to get protein isolation for further analysis, which in case you would attempt to probably have the highest expression levels possible? Are you trying to look at direct growth assays measuring root length or hypocotyl length or some sort of phenotypical output? In that case, you would probably want expression levels closest to your wild type level possible, so that way you can adequately compare the lengths to each other. If you're looking at fluorescence microscopy, you'll probably want a balance of these two, as the aforementioned reasons of silencing or abnormal protein function with overexpressed constructs could lead to an inaccurate output of fluorescence signal. So you want to balance having too much protein with too little, and many more. So in conclusion, you'll just want to make sure that your expression levels reflect what your end goal is for your experiment. With growth variables and conditions, once again, you do want to also ask what kind of goals are you trying to achieve. Are you trying to mimic wild-type conditions as closely as possible so that you're able to get something that would be seen in nature itself? Or are you just trying to isolate a specific variable to see whether or not that will change what kind of output you get? And so you want to identify these goals and make sure your experiment reflects that. And so what that means is your control variables should be level across. And one major control that I've actually seen is the amount of light that a plant receives in a growth chamber, pictured here on the bottom right. So what I found actually is that in growth chambers that I use, even though all lights would seemingly be at similar light levels, I noticed that different racks or different levels within my growth chamber would be exposed to different amounts of light for whatever reason possible. Maybe the reflection within the chamber causes certain levels to get more. Maybe certain light bulbs are older or newer than other ones, which could affect how much light your plants are getting on certain racks, and many more. So that's one thing you want to consider when introducing plants into a growth chamber. And a way you can actually make sure, or at least try to monitor light levels within different spots of your growth chamber is through the use of a light meter. And what a light meter will tell is pretty much how much light a certain point within your chamber is getting. And so that way you can make sure that's uniform across when you're growing plants in that chamber. If you're growing plants in a controlled environment, such as artificial media or different types of soil composition, you want to make sure that the soil composition is even across all your transgenic lines. And you'll also want to maintain that the media composition is prepped in the same manner across, because slight variations in concentration of nutrients in either can actually have a big effect on the phenotype seen in your plants. So these are just a couple of things you want to keep in mind when you're planning your growth assays. And lastly, with growth assays, you'll want an accurate way to quantify your phenotype and compare your wild type to your transgene or transgenic lines to each other. So you want to make sure that your output is quantifiable. And so what I mean by quantifiable is, for example, here you see that these are seedlings grown on a artificial media in light. And so let's say I'm trying to quantify the root length. Well, this actually would not be the best way to quantify root length, because in order to accurately measure my root length, I would actually have to pull out each of these seedlings individually, thereby, and since the roots are actually sensitive to any sort of physical handling, they could actually tear the roots and cause inaccurate measurements of your root lengths and so on and so forth. So you just want to make sure that your setup will help you accurately quantify your phenotype and output. Furthermore, you also, once again, want to make sure that your wild type and your transgenic plants are grown in equivalent conditions. And lastly, just to go into a little bit of my specific research experience with analyzing signaling pathways, there are a couple things you'll want to consider when doing this. And what I have pictured here is actually a hormone signaling pathway for the hormone ethylene. My specific protein of interest was actually IN2 and CTR1, which are both localized to the ER, CTR1 being upstream of IN2. And what we were seeing was that in the absence of ethylene, CTR1 was actually able to phosphorylate IN2 due to its kinase domain activity. And then in the presence of ethylene, this phosphorylation was inactive, allowing IN2 to signal downstream. So anyways, just a couple of quick things to consider when researching signaling pathways. You'll want to look at both in vivo and in vitro ways to look at post-translational modifications and signaling pathways / protein interactions to best tackle your research experiment. And I'll explain what I mean by this in the next couple of slides. And also you want to keep in mind the various ways that proteins can interact and transmit information downstream. So with in vivo versus in vitro techniques, my main area of advice would be to start small and then work your way to eventual in vivo confirmation. And so what I mean by starting small is, this is actually a figure from the same paper that I was a second author on. And what you'll see here is that we actually mapped out the domains for both CTR1 and IN2. And we actually found that since we knew that CTR1 was upstream, and we knew that it also had a kinase domain. So naturally we were thinking, well, maybe the way that CTR1 might propagate its signal downstream would be through either interaction with IN2 or actual phosphorylation and use of its kinase domain on IN2. And so next we decided to do a yeast-2 hybrid assay to see whether or not these two domains would actually interact, which we actually found interaction as evidenced by figure B. And figure C, we also found some level of confirmation that they interacted in a natural plant cell by transfecting tobacco leaves with a BIFC method, and we saw fluorescence when they were transfected into leaf cells, indicating that they also interacted within tobacco leaf cells. We were also able to do, in figure D and E, an in vitro kinase assay involving the kinase domain of CTR1 and also the hydrophilic domain of IN2. And in this way, we actually saw phosphorylation by CTR1 on IN2. And so we were actually able to confirm all of these techniques in vitro methods to confirm the fact that CTR1 was actually acting on IN2. And all of this occurred before we actually did any in vivo confirmation. So once again, what I would advise is to start small and then work your way up into vivo confirmation. Thereby, with So with the inexpensive methods such as these two hybrid assays, you're able to save yourself some time and money before you actually get into the more expensive research experiments. Lastly, I kind of want to go over real quick the various ways that proteins can actually interact and transmit a signal downstream. There are a number of ways that they can actually accomplish this goal. One of the main ones would be, and one of the more obvious ones would be protein-protein interaction, and also through this interaction, the recruitment of other proteins. And so just by naturally having for example, with my two proteins of interest, the protein signaling or the propagation of the ethylene signaling could occur just by natural protein-protein interaction, causing possibly some sort of abnormal localization of our protein, resulting in it being in a different area, propagating the signal as a result. It can also result in the recruitment of other proteins after these two proteins interact. So both of these are actually possible results from our experiment, which we actually didn't see, but could have happened. Secondly, we could have post-translational modification is also another method in which the signal can be propagated downstream. So in our case, we saw that the signal is actually propagated due to phosphorylation, but there are a lot of other post-translational modifications capable of propagating a signal. So that's something to keep in mind. And the best way to know whether or not this is a possibility is by analyzing your upstream protein domains to see whether or not there's a kinase domain of some sort of enzyme domain, that enzymatic activity that could propagate a signal. Lastly, and one of the lesser known methods of propagation would be protein cleavage and subsequent translocation of this cleaved protein. And so what I mean by that is with interaction between or phosphorylation of ion 2, you can actually see protein cleavage and subsequent translocation to the nucleus. And so that's something to consider, that maybe your signal isn't being propagated by the protein as a whole, but maybe only a portion of the protein is actually moving in response to a signaling stimulus. So the three of these things are the main things, what I would think would be the main things to consider when analyzing a signaling pathway. So that's pretty much a rundown of what of my experience with plant biology research and some of the pitfalls you might run into when conducting experiments in plant biology. And now I kind of want to go into a technology highlight of codon optimization, one commonly overlooked tool that could be very useful in the context of research in plant biology. So one common thing that is encountered when attempting to isolate protein or looking at protein localization through the use of fluorescence microscopy is that natural levels of expression are not enough to produce an adequate signal to see with your camera of choice or microscope of choice, and also it might not lead to enough yield for protein levels for downstream experiments. And codon optimization can actually counter this. So what exactly is codon optimization? It is the process of optimizing a transgene to achieve the highest possible levels of productivity in any given expression system, whether the sequence is naturally occurring or recombinant. And one thing you'll want to keep in mind is this is actually different from protein design. So protein design actually alters the amino acid sequence of your protein of choice, while codon optimization just uses synonymous mutations to introduce a more favorable codon of choice. So why do actual codons matter? And here are some of the facts as to why they actually matter. We actually see a high level of redundancy in the genetic code. And you can actually see this with the codon table here on the right for RNA or translation. And so you can see that there are upwards of 4 codons that can actually translate into the same amino acid. However, what's often overlooked is that synonymous mutations with these amino acids, and by synonymous I mean any mutation that changes the actual codon but doesn't cause a change in our amino acid, can actually affect protein expression rates up to 1,000-fold. Synonymous mutations themselves can alter protein conformation, stability, and function. And so codon optimization actually plays onto this by introducing synonymous mutations that will favor efficient soluble protein expression in your host. Some evidence for codon optimization, or some of the variables that you need to consider, are listed here. And one of the main ones you can see in the middle would be codon uses bias, which is actually the fundamental tool used for a lot of codon optimization engines and tools out there. But there are also a lot of other variables that need to be considered, such as GC content, internal chi sites and ribosomal binding sites, codon context, and RNA secondary structures, to name a few. And so you'll want a codon optimization tool that will take all of these into account to give you the best optimized sequence possible for your gene of interest. And also, you'll want flexibility to adjust the weight of different factors and or add customized constraints. And here at GenScript, we actually have a Optimgene codon optimization tool that is ideal for this. And so with our tool, you're able to filter out specific restriction sites for any downstream applications you might have. You can actually reduce similarity between library members if you're constructing a gene library. And you can actually use alternative codon tables and condition-specific codon preferences, all aimed at tailoring your codon optimization experience towards your specific experiment. So these are all things to keep in mind, and the GenScript's Optimgene tool can help you reach these goals. So a little bit of background on our tool itself. We actually have what's known as a particle swarm optimization algorithm patented with our Optimgene tool. And what this does is it's actually able to simultaneously consider all of the aforementioned variables at once when optimizing your sequence of interest. And when you're able to consider all of these variables, you see a much better optimized final product than if you were only to consider codon usage bias in your optimization process. So what we have here are images or gel images of the production of proteins with single domain or subunits expressed in various hosts. And you can see that with particle swarm optimization by Optimgene, you're able to see an increase in expression in E. coli, P. pastoris, insect cells, and mammalian cells. All these as a result of our Optimgene platform. And looking at our Optimgene platform versus our competitors, you can see that in 92% of cases, we see an either equal level of expression or an increased level of expression. And in only 8% of the cases, we actually see a decrease. So overall, our proprietary platform actually will either equal or outcompete our competitors' optimization tool. Furthermore, we also have the experience behind our Optimgene platform. We have over 500,000 sequences that have been optimized by GenScript and counting in all major expression systems, including plant. So right here we have listed that about 6% of this number is associated with plant optimizing projects, and that number is always on the rise. So that's something to keep in mind that when using a specific codon optimization tool. Also, GenScript has actually delivered over 5,000 proteins in 4 expression systems, and statistics show that there's a 95% success rate for all of these protein projects. So not only do you have a optimization tool that considers all these multiple variables, but it's also a proven effective tool for increasing or optimizing your gene expression. So all these facts, and as you can see here, we have hundreds of papers that will also cite GenScript for our codon-optimized gene synthesis results. And all these facts kind of lead to the idea of, well, why would I want this for plant biology? And the simple answer to that is the fact that if you really consider the amount of manpower or maybe resources needed to not only PCR a specific gene out of a plant, such as Rhabdopsis, mutagenesis and resulting cloning procedures that need to be done before you get your specific plasmid of interest, codon optimization or gene synthesis and subsequent codon optimization is an attractive option in that you get a product that is not only expressing at the highest levels possible in your host system, but also get 100% sequence guaranteed. And so you save yourself a lot of troubleshooting time and resources, and you're able to get to your experiment or whatever purpose you're using downstream more quickly. So just a quick rundown of what GenScript has to offer in terms of gene synthesis. We have a lot of gene synthesis options to fit your research needs. We have our standard custom gene synthesis. We also have various rush and fast gene synthesis options in case you need your construct a little bit quicker. We also have high throughput gene synthesis if you're ordering 25 genes or more to make it more economical to order such a high volume of genes. And we also have our GenScript platform to synthesize longer genes. And so as you can see, we have a lot of options to fit whatever your research needs might be. We also offer a variety of downstream bundling services, and one I want to highlight in particular is our express cloning service. We have over 151 free vectors offering competitive pricing and turnaround times. And also, if you don't see your vector of choice listed here, we can actually take your custom vector for a little bit, for just a minimal fee. And so this actually offers you a high degree of not variability, but a high degree of flexibility when planning your experiments. We also offer things such as vector arc storage of any completed plasmids you might have, express mutagenesis for your gene synthesis project, and also plasmid DNA preparation services. So overall, if you're able to bundle all of these into one research workflow, it can actually save you a lot of time in your plant biology research endeavors. Just to wrap up, GenScript, we're actually one of the most cited biology CROs. We not only have gene services, but we also have peptide services, protein services, antibody services, DB services, and also catalog products. And as you can see, all of these would lead to a degree of flexibility when planning your research experiments. So keep that in mind the next time you're planning your research. That's it for today. If you have any questions or feedback after today's webinar, you can email me at david.lin@genscript.com. Also, our webinars are all archived at genscript.com/webinars.html. And if you need any more information on our gene synthesis, codon optimization, or any other molecular biology services, you feel free to email us as well as visit our website at www.genscript.com. And so now I'm going to open it up to any Q&A, any questions you might have about anything that was presented today. Let's see here. Okay, so this is actually an interesting question. I actually got a question regarding BIFC. So the question states, what exactly is the process for using BIFC in terms of plant experiments? That's a very good question. You can actually use BIFC, and I'm going to speak with my experience in terms of Arabidopsis. So you can actually use BIFC both transiently and stably expressed. The only caveat with stably expressed use of BIFC is that the expression levels tend to be a little bit lower, so your chance of actually seeing protein interaction is lower. If you're actually able to use transient expression, and by transient expression I mean manually injecting your agrobacterium containing your constructs into the underside of, say, a tobacco leaf, while it is transient, meaning that it won't last for very long, in this case a couple of days, the expression level is actually a little bit higher, so that way you're able to see, you have a much higher chance of seeing interaction of your two proteins. So those would be the two methods I would recommend for using BIFC in your experiments. Let's see, what else do we have? How exactly would I go about using codon optimization in my experiments? That's a good question. So there are tools out there that can actually give you your optimized sequence once you input your gene of interest in them, but how would you actually obtain that sequence? And that would go back to using some sort of CRO such as GenScript to artificially synthesize your gene of choice. And in that way, you get a product that is going to be expressed at the highest levels possible in any host system. And that way you can get to your downstream applications a little bit quicker, such as fluorescence microscopy or protein isolation. So that's what I would recommend, is doing a little bit of research into any sort of gene synthesis tools that you might want to use to obtain your codon-optimized gene. I got another question now. Should one optimize the transit peptide encoding sequence for expression of a transgene in plants? Transit peptide encoding sequence for expression of a transgene. I don't think I actually understand the question. What I think you mean might be, so in terms of codon optimization, it optimizes your CDS of your gene of choice. So there's not actually any optimization of your vector, if that's what you mean. I'm actually not quite sure what you mean by transit peptide encoding sequence. If you want, feel free to email me after the webinar is over, and I can, with some clarification, and I can have a more in-depth discussion with you about this. But as of now, I don't think I'm actually answering it accurately, since I don't think I understand the question. So yeah, feel free to email me with that or any other questions you might have when the webinar concludes. And once again, my email is david.lin@genscript.com. I guess that's all the questions for today. Thank you all for attending, and I wish you the best of luck in your plant biology research.